Well, Professor of Vaccinology at the University of Witwatersrand, Shabir Madhi joins us now. He's also the trial head for the Oxford vaccine study in South Africa, and he joins us via Skype. Thanks so much indeed for joining us, and welcome to the program.
Good evening. Thank you for having me.
All right. So, how excited are you about this trial stage of this vaccine?
So, I mean, it's obviously a privilege to be involved in terms of the clinical development of this vaccine, as well as the vaccine that was developed at the University of Oxford. Both of these vaccines are very different in terms of their construct, and in a way that is good for us because it sort of probably improves our chances of showing success for at least one of the two vaccines, if not both vaccines.
All right. Explain to us the difference between the two and how they are meant to work.
So the University of Oxford vaccine is using a relatively new technology when it comes to the field of vaccinology. In fact, there's only one other vaccine that's been licensed using that particular technology, and that is a vaccine for Ebola virus. The manner in which that vaccine is constructed is that it takes a virus which is known as adenovirus, which doesn't really cause severe illness in humans, and that is genetically engineered in such a way that the spike protein, which is found in the SARS coronavirus 2, the genetic material of that is inserted into the other virus. And eventually that is presented to the immune system to mount an immune response against that spike protein. The Novavax vaccine takes a more traditional approach in that it actually takes the spike protein itself. It formulates it in such a way that it's presented to the immune system to enhance the immune system's ability to produce antibody, as well as what we call T-cell immunity, which is induced by this particular spike protein. So they're sort of different constructs, but at the end of the day, both of them are trying to achieve very much the same sort of outcome, and that is to stimulate the immune system to produce antibodies, as well as to induce what we call T-cell immunity, which is another component of the immune system that's important when it comes to protecting against COVID-19, as well as preventing infection from the virus itself.
All right, so this Novavax version, give us a timeline that you estimate might start to show us something. I mean, to get to the trial stage, you've already seen something, haven't you?
Well, certainly. So with the Novavax vaccine as well as the University of Oxford vaccine, both of those vaccines have already completed what we call phase 1 studies, which is where they evaluate it in a relatively few number of subjects, less than 100 subjects usually, or participants. And we look at the safety of the vaccine, at least for very common side effects, as well as the immune responses to the vaccine. So both of those vaccines have gone through that sort of initial phase 1. Now we're going into a phase 2 study, which involves enrolling up to about 1,000 or 2,000 participants. In our instance, we're enrolling 3,000 participants. And what that is really geared towards is getting broader repertoire readout in terms of the safety of the vaccine, as well as whether the vaccine protects against COVID-19. Now, the question that you asked in terms of when will we know, that is really the golden question. And to some extent, that depends on when people actually get infected with the virus. So the way these studies are conducted is that people are randomly assigned to receive the vaccine or a placebo substance, and neither the investigators nor the participants will know which arm they've been allocated to until there comes a point in time where we've accrued a sufficient number of individuals that have developed COVID-19. And at that point, we can do an analysis to determine whether the vaccine actually does protect against COVID-19 or not. That time point really depends on how quickly people get infected with the virus, which is something that is obviously beyond the control of the investigators in these studies.
All right, you've mentioned a number of things which piqued my interest, and the one is waiting for people to get the virus. And we're working very hard to slow it down, to stop it. Is there a danger that you don't get a critical mass of people infected in time?
Well, like I said, surely that is I wouldn't call it a danger. I mean, we don't wish for people to get infected. We need to be clear about that. All it really means is that it will probably take us longer time to get to an answer compared to people to get infected very quickly. It's very unlikely that we're not going to see another outbreak of COVID-19, irrespective of whether we're doing a vaccine study or not. The big issue is, when is that next wave of the outbreak going to come, and what is the magnitude of that going to be? I'm hoping that the next wave takes as long as possible and that it is much less severe than the one that we just currently experience. But all that it will mean for the study is that it will delay in us being able to address the question as to whether this vaccine actually protects COVID-19 or not.
All right, and we're talking about vaccine, but given what it does, what it tells the body in terms of if you see this virus and it looks like this is what you must do, can we not deploy it perhaps as part of treatment? No, so vaccines are a very different approach in that it takes some time, up to about 2 weeks to up to 4 weeks, and some instances, you need to at least give 2 doses of vaccine before the body's immune system is adequately stimulated to produce its antibodies, which basically prevents a person from getting infected. Now, another version of this is what we call a monoclonal antibody. So even serum therapy as an example. That is something which has recently been in a public light in South Africa. And that uses a similar approach, but in that instance, we're passively administering that antibody directly to the individual. That antibody is probably going to last for a much narrower period of time. And in fact, just today, there was a publication that came out, which in fact indicates that the use of the serum therapy, which is basically antibody that's included in the serum product, actually has reduced death from COVID-19. So that is a very different approach. It is sort of aligned with a vaccine approach, but is actually giving the antibody itself rather than wanting the body's immune system to develop the antibody. All right, so in terms of vaccines generally, you know, some of the ones that we get for, I don't know, BCG and TB and so on and so forth, you get a couple of shots and that's you done for life. These vaccines that are coming through, will we be getting them like flu shots every year, every 2 years? What's the likelihood of these vaccines?
Right, so the reason we receive flu shots every year is because with the flu virus, what happens is that it is what we call antigenic drift, which means that a flu virus undergoes some mutation, and hence the vaccines that are used in this year might not necessarily work as well next year when there's a mismatch between the strain that's included in a vaccine compared to the strain that's actually circulating. Fortunately, with the SARS coronavirus 2, although there are mutations that are arising, those mutations don't seem to be too important in terms of the component of the virus which is largely being targeted by this vaccine. So unlike flu, with this virus, genetically it's much more stable, so that wouldn't be a reason for annual vaccination. But the big unknown with these vaccines, with SARS coronavirus 2, is how long will the immunity that's induced by the vaccine last? And certainly that would be an ongoing question that needs to be addressed moving forward. Based on what we're seeing with natural infection, as well as experience with other common cold coronaviruses, which have been around for decades now, it seems that at least the T-cell component of the immunity, which is important in terms of clearing the viral infection at an early stage, it tends to last for a number of years. So hopefully we won't need revaccination each year, but I think what we would require, at least at the start, is 2 doses of the vaccine spaced about a month apart.
All right. You know, in the past we've seen SARS, we've seen MERS, now COVID-19. Are we getting a better understanding of coronaviruses generally so that next time if there's a COV-3 that comes through, we might be able to act quicker and recognize it quicker?
Yeah, so I think the lesson that we've learned with MERS, SARS, and SARS coronavirus 2, these all belong to the family of coronaviruses, but they behave very, very differently, both in the manner in which they stimulate the immune system as well as the type of illness it causes. So although they belong to the same family of viruses, it's difficult to tell whether we're going to be any better prepared when the next coronavirus outbreak occurs. It depends on how sort of similar it is to the viruses that we've already experienced. So unfortunately, it's really difficult. But hopefully, I think the important lesson this time round, and what has been shown with vaccines, is that, as you know, there's close on to 40 vaccines against this virus that are now in clinical trials, not even 7 months since the first discovery of the virus. So that itself is a huge, huge achievement by the scientific community to go so fast to be able to at least get these vaccines into clinical trials in humans.
All right, so 2,900 people, South Africa. How are they selected? Is this sort of to try and match the demography of the country, or people that are more vulnerable given what we've seen and the science that's there at the moment? How do you choose the 2,900 people?
Right, so we're wanting our vaccine studies, the results of the studies, to be as generalizable as possible. So we try to place as few restrictions in terms of who can participate in the vaccine study. And obviously, the selection is largely based on who eventually volunteers for participation. So the starting point here is that we're wanting to rate individuals between 18 and 64 years of age. And even though we do include individuals that have got underlying comorbidities such as hypertension, diabetes, those conditions need to be relatively well controlled. So this particular study probably is very different from other studies that will be at a similar stage of clinical development, in that at a very early stage, we're wanting the population group that's involved in the studies to be what would otherwise exist in the general population. So the study is open to all volunteers between 18 to 65 years of age. And if anyone is interested, they could go to the University of Witwatersrand website and they could actually do a screening to determine whether or not they're eligible and where they might be able to enroll into the study.
All right. And when does it kick off? When must they register by? All right, I think we might have lost.
Sorry?
As I was just saying, when does the trial kick off? When must people have registered as volunteers by?
Well, we're going to continue enrolling until we reach the 3,000 participants. So I think there's still ample opportunity.
Okay.
We expect to enroll the 3,000 participants roughly by the middle of October. So between now and then, there certainly would be opportunity for anyone that's willing to volunteer to participate in the study to come forward.
All right. That's where we're going to leave it. Thank you so much indeed for joining us, and good luck with this second trial that you're involved with.
Thank you.
Thank you so much.
Very much.